Hi everyone, how are you? Good to see you back and happy Sunday. It's a really nice day today, so I hope you're all having a good weekend. I'm actually on nights this weekend, so I am going to go to sleep after this. But yeah, thankfully it wasn't like too bad last night, so yeah, we'll just give everyone a couple more minutes and then get started. Let me know how your week has been. How was revision? Have you covered things that you wanted to, or is it getting a bit difficult? Let me know in the chat. I'd like to hear how you're doing. Yeah, just wondered how everyone was getting on with their kind of revision plans and if anyone started doing questions and things yet. We'll get started in a moment. So today we're doing the first half of neuroscience. So this is one of the bigger topics in the syllabus, and it comprises 25% of the exam, which is 37 or 38 marks, which is quite a lot of marks, but is a very, very big topic. So today I'm going to focus as usual on the high yield things. I'm not going to cover everything. I'm just covering the main things that I think are really important for the exam. There are some things obviously that are just really sort of out of the scope of being able to fit that in, but these are the most important things that I think you definitely need to know. And we've split it into two halves anyway, so hopefully we'll be able to cover good chunks of the material, and then after that we'll finish part 2 and then we'll do molecular genetics. Molecular genetics technically just part of basic neurosciences, but because it's its own quite big topic, it's often separated in revision sort of structure, so I have separated it as well. So today we'll do the basic techniques in neuroscience, cells, neurotransmitters and receptors, and some neuroanatomy. So just to start off with, we'll start with the boring stuff, which is the action potentials. So the main things to know about this is the kind of numbers really. So remembering that the resting state is -70, and then for the action potential to, oh, the printer's going off, the membrane potential has to be raised to -55. Let me just turn that off. I just printed out all the notes from the talk, so the printer's overheating. So yeah, I think raising the membrane potential being raised to -55 is again another really important number. And then the other important number to remember is at 40, the sodium channels close and the potassium gates open. So that's when the membrane potential falls back again. There's not really much else that can be asked in a question for action potentials apart from those three things. So then we're moving on to EEG. So I'm sure you all know what EEGs do and what they are for. Main things to know is that they can be affected by psychotropics. They can also be triggered by activation procedures such as hyperventilation, photic stimulation, which is kind of like flashing light stimulation, certain drugs, and sleep deprivation. So they sometimes will do that on purpose to bring on a seizure, to see what kind of seizure it is and look at the EEG and look at the types of waves in there so they can analyze it. Another thing to note that usually it's used to kind of rule out organic conditions. In this case, things like epilepsy would be classed as organic. And then, you know, considering things like what used to be called pseudo seizures, but now is psychogenic non-epileptic seizures. And then in meditation, you get increased alpha and theta waves, and also in very experienced meditators, you get gamma waves too. So in terms of medications and drugs that affect EEG, lithium increases all of the waves that you get in EEG, and lamotrigine decreases them. So this is just a kind of summary of that, which we can go through. So I try and remember the order of the numbers with this DTASBG. So that's delta is 1 to 4 hertz, theta is 4 to 8 hertz, and then alpha is 8 to 12 hertz, sigma 12 to 14, beta 12 to 30, and then gamma is 30 to 100. It just kind of helps with remembering in questions. So sometimes this can be at EMQs, and they might ask which is the correct numbers for the type of wave that you're looking at. So with alpha waves, they're found when people are relaxed, and usually when their eyes are closed. And they can be kind of things like people do hypnosis, that can be an alpha state, and in meditation. Then beta state is when you're busy concentrating, so there's quite a change from the frequency from alpha, if you can see in the picture. And then theta is more like drowsiness, some medication, also some meditation. But if this is seen when awake, that indicates pathology, which is one key point to note. Delta waves, so this occurs frontally in adults and posteriorly in children, is part of slow wave sleep. And again, this is also pathological if present when awake. Then sigma are like sleep spindles, which are kind of bursts of these activity in stage 2 sleep with K complexes, and you only get that in stage 2 sleep. So it's quite a specific wave to see. And then the loss of sigma waves is a feature of familial fatal insomnia. And then gamma has got no specific areas necessarily, but is seen in advanced meditators. Okay. And then looking at pathology, so in normal aging, so in the older adults, diffuse slowing is normal, but there can be focal slowing in the left temporal region as well, which is classed as part of normal aging. In delirium, you get a diffuse slowing as well with decreased alpha, increased theta and delta. In encephalopathy, there's diffuse slowing. DT, you get a fast hyperactive trace. Huntington's is flattened with no alpha waves and a low voltage ECG. Alzheimer's has reduced alpha and beta and increased delta and theta. And then the CJD sporadic type gets non-specific slowing early on, then later biphasic and triphasic synchronous sharp waves on a slow background with rhythm. And then in terms of seizures, so I put that picture of the penguins because it's, you know, that smile and wave, boys. So I think of that for spike and wave. For absent seizures, that's a really typical thing to be asked. So spike and wave is for absent seizures. So you can get generalized 3 hertz spike and wave with typical absence, and then you get slow generalized spike and wave with atypical. And then with focal is focal spikes, myoclonic is generalized 3 to 6 hertz polyspike and wave discharge. And then with generalized tonic-clonic, often it's difficult to obtain the EEG properly, and you can get a lot of artifacts in it, but it's fast rhythmic spikes and kind of slow waves as well with the jerks. And then there's the post-ictal period too. And atonic are drop attacks, which are generalized spike and wave again, and atonia at the time of slow wave. So I think just important to remember for seizures is just this whole spike and wave thing, which kind of is in seizures mainly, but that's kind of one of the characteristics of it. So spikes can be focal or generalized across widespread regions of the brain, and then the rhythmic recurrence of spikes is followed by the slow wave, is what's known as a spike wave. So I got a bit confused myself when I was looking at spike waves and spike and wave, and they're basically the same thing. So that's just terminology that is used. So now we'll have a look at some neuroimaging. So CT scanners are the ones shaped like a polo mint. They use ionizing X-ray radiation. For dense tissues like bone, they become white, and the scan is quite quick, and the quality is reasonable but not as good as an MRI, which is the next one, which aligns hydrogen ions and uses a radio frequency. And then this is detected by the machine once the atoms realign. And you get two types, which is T1 and T2 weighted. And there's also the perfusion MRI, which looks at the blood flow in capillaries. And then there's the functional MRI, which uses the oxygenation of blood to show the image. And then PET and SPECT both use radioactive substances and detect gamma rays and give an image from that. MR spectroscopy doesn't necessarily give an image of the brain. It looks at specific metabolites in terms of spectroscopy, so looking at different parts of chemicals that are in the brain. And then DTI is a diffusion tensor imaging, and that looks at the paths that water takes as it diffuses through the white matter and shows the white matter tracks. So I'm just going to show you a few pictures now, and I want you to guess what each of these show. So if you can just maybe message in the chat what you think they are. So what do you guys think that this one is? Great. So some people are saying, yeah. Okay, so on the right, so maybe don't focus on the part on the right of the actual brain pictures, maybe have a look at the kind of shape of that graph, and kind of out of the ones that we've looked at in the table, think of which one it would be. So this is the magnetic resonance spectroscopy, which is MRS, and that allows the chemical sampling of the brain. It doesn't produce an image, but gives a breakdown of the amounts of different chemicals, and it looks at things like Noradrenaline, acetylcholine, glutamate, and all kind of work out how much is in there. So that is the MR spectroscopy. Which one do you think this is? Yeah, okay, so different thoughts on this. I feel like this is the coolest out of all of them in terms of brain scans. This is actually the DTI. So what you're looking at is the white matter tracks that are in the brain. And the way that it does this is it's a type of MRI that looks at the way the water moves through the tracks. So it kind of comes up in that way, but they weight it all differently, so it's different colors, and I think it looks really good. So that's DTI. Which one do you think this is? Come on, guys, I want your answers. Some people have given up. I don't want that. I want some guesses at least. It's fine to get it wrong. Yeah, good. Someone said DAT scan, maybe. That's a really good guess there. Okay, it's any other thoughts on what it could be? Okay, so this is actually a PET scan. So this is obviously the single photon emission, and it uses radioactive substances that are detected as well, but injected. SPECT detects the gamma rays as well as PET. So when the gamma decays, it gets detected, and then that produces the image. So that is a PET scan. And then this next one is a SPECT scan. So they're quite similar. They use a very similar style to collect the information, and it's both radioactive. So good guesses from everyone. I think you guys probably all know what this is. Is this a CT or an MRI? Okay, so some people are saying CT, some people are saying MRI. So this is an MRI head, and the reason we can tell is because the next image is the CT, and there's just a different quality to the image. With the MRI, you're looking at you're just looking at a clearer picture. You've got more detail, more outline, and then you can also weight it T1 and T2 for the signal for liquid. Whereas in the CT, it's quicker. You do get a fairly good quality image, but the denser tissues like bone attenuate the most, and they appear white. And then obviously the air appears black. Okay, great. Well done, guys. What do you think this is? Yeah, some interesting thoughts. Yeah, and some people are saying another type of MRI. So that's correct. So this is a functional MRI. And this is basically looking at the fact that blood has different levels of oxygenation, which produces different signals. So when they're doing fMRI, they're looking at the amount of oxygen consumption in particular areas. And so this technique is called blood level dependent or bold contrast. And this can be used sort of in research and things. And lastly, this is another type of MRI, which is called pMRI, which is perfusion MRI. And this looks at the microscopic blood flowing capillaries. It's not really, it's kind of mentioned as a side note in the syllabus, really, but it is part of this topic. So just good to cover everything. So any thoughts on those? Let's have a look at some cells now. So breaking it down to looking at what's inside a neuron. So inside a neuron, it's got the same things as what other cells have. So it's got mitochondria, it's got a nucleus, it's got Golgi apparatus, microfilaments and microtubules, lysosomes and melanin. And the cell body, which is the soma, makes up the main part of the neuron, and then the axon is the kind of long part as well. So that comprises a normal neuron. And then melanin, I always thought of as kind of like it protects the DNA, but there's sort of other things that have been written that just say that it's a possible byproduct of neurotransmitter synthesis. So types of cell in the nervous system. So first of all, we've got the microglia, which is the top right. And these are the phagocytes of the CNS. So they will come and eat up all of the debris and kind of kill off viruses, any kind of bacteria. They will do the job of what basically a white blood cell does in the normal body area, but they'll do that in the CNS. And then astrocytes are there for structural support of the brain on a small level, and they remove excess potassium, they help form the blood-brain barrier, and they repair physical damage as well. So they've got these kind of long arms and legs, and that's the blue one on the right. Then the Schwann cells are the cells that produce myelin in the peripheral nervous system, and acoustic neuromas are formed from Schwann cells. And when people get Guillain-Barré, it affects this cell, and that's why they get that kind of demyelination. The ependymal cells are quite important because they line the inside of the ventricles in the brain, and they help produce the CSF along with the choroid plexus epithelium as well. And then lastly, we've got the oligodendrocytes, which are producing myelin for the CNS. So you've got Schwann cells producing myelin in the PNS, and then oligodendrocytes producing myelin in the CNS. And in multiple sclerosis, those are the cells that are affected. And now we'll have a look at glial cells. So these are just separated these are just the same cells, but separated into their own category. So the macroglia, which are the bigger ones, the astrocytes and the oligodendrocytes. And then you've got ependymal cells and the microglia as well. So just worth remembering what each of those does, as that comes up and it's easy marks. So as said before, the CSF is made by ependymal cells in the choroid plexus, and the lateral third and fourth ventricles can make around 500 mils a day, but because there is a reabsorption constantly of the CSF, there's actually only about, I think, 100 mils or so, that's 100 to 150 mils that's actually normally present, because it's always being reabsorbed. And the normal intracranial pressure is 5 to 15 millimeters of mercury. But when the pressure inside the brain increases, the absorption also increases, so that can affect as well. There's also a point to be made about comparing plasma to CSF. So the CSF has a composition which is very similar to the extracellular fluid in the brain, but is different from plasma. And so the main things are that the PCO2 is higher, which results in a lower CSF pH. So the CSF pH is lower than what you find in normal plasma. And the protein content is normally very low as well as glucose concentration being low. Chloride and magnesium tends to be higher. There's lower cholesterol, and also basically it's just kind of a different set of chemicals because of the requirements of the cells in the brain. So I got a question, I think I got this question in my exam both times, about mirror neurons. So one of them was asking whether it was related to empathy, which I got a bit confused about, because I think there has been some research done that does show like that they are related to empathy, but actually the main thing that they were originally researched for was part of a model for understanding imitation learning. So watching someone else do something and then learning how to do that thing, and they mirror neurons discharge in them. So the experiment was done with monkeys to find this, and their mirror neurons discharge when the monkey does an action and they see another individual doing a similar action. So not to be confused with other things. And then this is also another question that I got in my exam, which said, how many parts does a G protein coupled receptor have? And I actually put 6, but there's 7. And it's kind of called a serpentine formation. So it's just worth remembering that there's 7, and metabotropic receptors use G proteins. So that's just something to think of. Okay. Now we'll talk a little bit about some neurotransmitters and receptors. So obviously neurotransmitters are how neurons communicate with each other and target tissues. They're synthesized and released from synaptic cleft from nerve endings, bind to receptor proteins in the cell membrane of target tissues, and that causes either excitation, inhibition, or some other kind of thing that happens. So neurotransmitters are classed into these 4 different classes. So they're all small molecules apart from the large molecules. So most of these are small molecules like acetylcholine, all of the kind of serotonins, amines, amino acids, and then carbon monoxide and nitrous oxide. And then the large molecules are things like cannabinoids, oxytocin, endorphins, somatostatin, cholecystokinin, and substance P. And I think the way to remember that they are large molecules is that they just seem quite big. If you look at their chemical structures, what makes up oxytocin or an endorphin, it's just quite a lot more like amine ends and kind of methyl groups and things on there that are larger, and so are more difficult kind of in terms of membrane crossing, whereas the small molecules are, you know, quick and easy most of the time. These are the excitatory neurotransmitters. So They are acetylcholine, epinephrine, norepinephrine. Dopamine has both an excitatory and inhibitory function, and then there's also serotonin and glutamate. And then the inhibitory neurotransmitters are, again, dopamine does both, but glycine and GABA is the main one. So now we'll talk about ionotropic receptors and metabotropic receptors. So ionotropic receptors, they act by opening an ion channel on the cell surface, and there's like a direct channel between the outside and the inside. When these are used, when these receptors are activated, the effect is usually quite short-lived, and it's on a very specific action. And fast-acting neurotransmitters bind directly to ligand-gated ion channels. So the neurotransmitters with ionotropic receptors are GABA A, glutamate, which is excitatory, glycine, which is inhibitory, serotonin, only 5-HT3, and then acetylcholine nicotinic. So they don't all have to be the same type. It's definitely worth remembering those 5, just to know that those are the ionotropic receptor neurotransmitters. And then the metabotropic receptors, so these use G protein coupled receptors which initiate a reaction in the cell, and there's a longer lived effect and it's more diffuse. So this is more of a slow kind of reaction from the metabotropic receptors, and these slow acting neurotransmitters bind to G protein coupled receptors. And again, it's worth just having a read of those and noting that GABA A does ionotropic, whereas GABA B does metabotropic here. And then serotonin, the rest of the receptors, apart from the 5-HT3, are using ionotropic, and then, 5-HT3 using ionotropic, and then the rest are using metabotropic. Are there any questions on that so far? Okay. So now we're looking at catecholamines, so the benzene group, benzene ring with 2 hydroxyl groups, terminal amine group, and intermediate ethyl chain. And here you're looking at dopamine, adrenaline, and noradrenaline. And the catecholamines are derived from tyrosine. Sorry, I still have this cough. So just talking about noradrenaline now. So this is also obviously known as norepinephrine, and it has an excitatory function. It's a class 2 amine, which is a small molecule, and it uses metabotropic receptors. It's important to know the process of creating noradrenaline. So it starts off with tyrosine, which is converted to L-DOPA by tyrosine hydroxylase, and then to dopamine by DOPA decarboxylase. Sorry. Guys, I'm just going to get my inhaler. I'll just be back in a sec. Hey guys, sorry, I think I might have long COVID, I don't know, but I feel like this happens every time I try and speak for more than like 40 minutes, but anyway, hopefully it'll be okay. And thanks everyone who messaged saying take your time, and I'm okay. I'm okay, I'm fine. It's more just annoying, I feel, for everyone, but it's annoying for me. So I think we're talking about the process of converting to epinephrine, so noradrenaline. And so it's important just to remember the key enzymes there, which are, sorry. Hey guys, sorry, I'm literally really sorry. You probably already read the whole slide by now, but basically noradrenaline is also broken down by COMT and monoamine oxidase as well. It's important to remember that it's synthesized in the blue spot, which is in the pons. It's increase in anxiety, which is a kind of skydiving picture, and it's decrease in depression. Serotonin is 5-HT, synthesized in the CNS in the RAFE nuclei, or RAFE, which is in the brainstem, and then also in the GI tract by the enterochromaffin cells. So this is decreased in depression and anxiety, and is synthesized from L-tryptophan in the diet, which is important because L-tryptophan can cross the blood-brain barrier, but serotonin can't. And then L-tryptophan is converted to 5-hydroxytryptophan by tryptophan hydroxylase, and then the L-aromatic amino acid decarboxylase enzyme converts 5-hydroxytryptophan to serotonin. So this is really like quite they're quite sort of difficult things to remember in terms of enzymes. So I'm not really sure of a good way of remembering these, if anyone has any ideas, but I think I just wrote them out and tried to remember each one and did questions on them. But don't feel bad if you can't remember all the long names, because there are quite a lot of them. Maybe just remember tryptophan, because tryptophan is found in things like milk and yogurt, and that can be recommended for like if people can't sleep at night, people say to drink milk, and that's because tryptophan can cross the blood-brain barrier and that can help with sleep, whereas serotonin can't. So maybe just remember tryptophan hydroxylase, and then remember L-aromatic amino acid decarboxylase, which converts that to serotonin. And then also the breakdown of serotonin is really important. So serotonin is taken up from the synapse by SERT, which is the what breaks down serotonin. So then monoamine oxidase then breaks it down further, and then aldehyde dehydrogenase, which is a bit of a weird one because you wouldn't expect that to be involved in serotonin, but it is. And that converts it into 5-hydroxyindoleacetic acid, which is 5-HIAA. And then the action of things like drugs are usually affected by the SERT. So amphetamines, cocaine, tricyclics, MDMA, and SSRIs block SERT, and that basically increases the synaptic amount of serotonin, which can be why it has such a good effect of making people feel good and that kind of thing. And then coming back to the serotonin receptors, we mentioned earlier that 5-HT3 is ligand gated, and it's ionotropic, and it's most associated with nausea as well, whereas 5-HT7 is associated with circadian rhythms, and 5-HT2 stimulation relates to SSRI side effects of insomnia, agitation, and sexual dysfunction. So the serotonin receptors are G protein couple receptors, apart from 5-HT3, which is ligand gated, which I think is probably the most important thing to remember about the serotonin receptors, apart from obviously 5-HT7 and 5-HT2. And as mentioned before, so with the breakdown, there is the breakdown of serotonin, there is this kind of molecule that is created, which is called 5-HIAA. And this was really important because it has a lower CSF concentration in people who have depression, in a third of people who have depression, and also having low levels of this linked to a higher risk of suicide and a worse response to antidepressants. So this often comes up as well, and increased levels of aggression are linked to this too. So dopamine, this was first synthesized in 1910 by James Ewens and George Barger, and its role as a neurotransmitter was discovered by Arvid Carlson, who won the Nobel Prize for it in 1958. And so it's originally made from tyrosine, which is found in food, so it's found from the diet and amino acids, and is synthesized in the ventral tegmentum, also in the substantia nigra pars compacta and the arcuate nucleus, which is part of the tubulo-infundibular pathway, which we'll go through soon. Dopamine's increased in Huntington's and in schizophrenia, and is decreased in Parkinson's and depression. Tyrosine is converted to L-DOPA by tyrosine hydroxylase, and then L-DOPA is converted to dopamine by DOPA decarboxylase. So these are the main enzymes for dopamine. So tyrosine hydroxylase and then DOPA decarboxylase. Dopamine, as you know, inhibits prolactin, which is why antipsychotics increase prolactin, because they decrease the dopamine, so then the prolactin is no longer inhibited. And then moving on to GABA. So GABA is synthesized in the nucleus accumbens, decreased in Huntington's and anxiety, and is a major inhibitory neurotransmitter in the brain. And there was some reason why I put Fox there, and I can't remember what the reason was. Oh yeah, I was Huntington's, so like hunting, so fox hunts. So it's decreased in Huntington's and anxiety, GABA, and it is the major inhibitory neurotransmitter in the brain. That's really important. And then the reason I picked a mountain for acetylcholine is because acetylcholine is synthesized in the basal nucleus of Maynard, and it kind of reminds me of like the base camp of a mountain. So just think of acetylcholine like at the base camp of a mountain. So that's the basal nucleus of Maynard. It's increased in Parkinson's disease and decreased in Alzheimer's and Huntington's. It's involved in memory and learning, and it's the main neurotransmitter in the autonomic nervous system. And it's formed from choline using choline acetyltransferase, and it is broken down to choline and acetate with acetylcholinesterase. So there are two receptors that are stimulated by acetylcholine, which are the nicotinic and the muscarinic receptors. So those are the two main types of acetylcholine receptor. I didn't realize that. I thought they were their own like receptors for whatever they do. So, but to be fair, nicotinic can be stimulated by nicotine and muscarinic can be stimulated by muscarine, but they also both respond to acetylcholine. And nicotinic receptors are ionotropic, and muscarinic receptors are metabotropic. So just a quick note on melatonin. So this is synthesized from serotonin in the pineal gland, and it's released more in dark environments, and it kind of regulates the circadian rhythm. So it can be given as well for mood. And now we're going to have a look at the hormones involved in appetite. So cholecystokinin, this is secreted by the duodenum, and jejunum in contact with food. It stimulates the gallbladder to release bile, stimulates the pancreas to secrete pancreatic enzymes, and acts on vagal neurons to signal satiety to the medulla. So this reduces appetite. So I find it interesting that it acts on vagal neurons. So that signals straight to the medulla oblongata from the vagus nerve. Ghrelin is secreted by endocrine cells in the stomach and increases appetite, and it acts on the hypothalamus. Leptin is made in fat cells, adipose tissue, and it reduces appetite. It's released when the person has eaten. And then neuropeptide Y is made in the hypothalamus, and it also increases appetite as well. Here's some more hormones. So somatostatin is secreted by gastric glands in the stomach, inhibits the release of gastrin and hydrochloric acid, inhibits secretin and cholecystokinin on the duodenum, and inhibits release of glucagon on the pancreas. Secretin is secreted by duodenal cells when exposed to acid, and this hormone actually causes neutralization of the acid because it stimulates the pancreas to secrete bicarbonate. And then gastrin is secreted by the stomach and the duodenum. It stimulates exocrine cells to secrete HCl and pepsin, which is an enzyme. So these all kind of work together in a sort of functional way when people are eating or getting hungry, and they can be affected by various other things, but these are the most important ones. So just make sure you know those for the exam. Okay. So moving on to the endocannabinoid system. So the endocannabinoid system modulates neuronal activity. It's involved in the regulation of cognition, metabolism of energy, inflammation, and sleep. So there are different neurotransmitter systems which are modulated by this, and they're all the kind of main systems, including like dopamine, glutamate, and GABA. So the two main receptors are CB1 and CB2, which are both G protein coupled receptors. CB1 works in the CNS, whereas CB2 is in microglia and in vasculature. So CB1 is probably the main one that actually has an effect on kind of the effects that people have from, using exogenous cannabinoids. The main ones that are made within the body are anandamide and 2-arachidonoyl glycerol, which is also known as 2-AG. So 2-AG is an agonist for both CB1 and for CB2, and anandamide is a weak agonist for both. So basically, impairment of the ECS after cannabis use has been associated with increased risk of psychotic illness, which whereas enhancement of the ECS with CBD, so the non-active, so the two parts of exogenous cannabinoids are THC and CBD, but using CBD has shown anti-inflammatory and antipsychotic outcomes in a study and in clinical trials. So there's obviously still a big debate about using cannabinoids in terms of safety as well as other things. So they had increased anandamide levels in the CSF and blood, and increased CB1 expression in peripheral immune cells of people with psychotic illness compared with healthy controls. And that was a study in 2019. Okay, so we can get started with some of the neuroanatomy before the break, and then we'll continue that after. So I kind of decided to mainly focus on the things that come up as questions, because I feel that learning neuroanatomy is quite, it can just be one of those things that you need to look at yourself to understand, and maybe not necessarily easy to get just from watching it. But I won't embarrass everyone by showing all the frontal lobe and all of those, because I feel that those most people know already. So I'm going to focus on kind of the things in the syllabus that are difficult to learn. So the white matter pathways and the neurochemical pathways mainly, but also some of the small things like the cranial nerves and some of the other smaller parts of the anatomy. But again, we will be covering that in the next section. We will look at things like the gyri and kind of like the aphasias and all of those kind of things, which come up in the next part. So starting off with our favourite, which is the cranial nerves. So I think one thing that is important to know is where it originates from. This is often missed out in revision. So this happens to be a popular question in the exam for some reason. But where does, like, for example, the olfactory nerve originate from? So that's from the telencephalon. And then going down to the optic nerve originates from the diencephalon, then oculomotor and trochlear from the midbrain, trigeminal, abducens, facial, and vestibulocochlear from the pons, and then the rest are from the medulla, so glossopharyngeal, vagus accessory, and hypoglossal. Olfactory nerve exits the skull from the cribriform plate. It's mainly sensory modality, and while it's purely sensory modality, you can't move your nose with the olfactory nerve, and the purpose is for smelling. The optic nerve exits the skull from the optic foramen. Again, is sensory, and this purpose is for vision. Oculomotor exits the skull. Superior orbital fissure is a motor modality. Does eye movement, pupil constriction and accommodation. Trochlear nerve comes out at the superior orbital fissure as well. Again, is motor and does the down and out eye movement. Trigeminal, so it has three parts to it. So the first one, V1, is the ophthalmic branch, and that exits from the superior orbital fissure. The V2 maxillary branch comes out at the round foramen, and the V3 mandibular branch comes out at the foramen ovale, or the oval foramen. And this has a mixed modality, so it has some sensory components as well as motor components. So the sensory components is that it does the sensation of the anterior 2/3 of the scalp, and also does motor things, so mastication, which is chewing, comes from the trigeminal nerve. The abducens nerve also exits the skull from the superior orbital fissure along with oculomotor and trochlear, and that has a purely motor modality and moves the eye outwards, so away, abducts the eye. The facial nerve comes out at the internal auditory canal, and that has a mixed modality, so sensory and motor. Sensory-wise, it does the taste for the anterior 2/3 of the tongue, and the facial expression for the motor aspect. Vestibulocochlear exits at the internal auditory canal as well, and that has a sensory component purely, and that only does hearing. Glossopharyngeal exits with vagus and accessory from the jugular foramen, and the glossopharyngeal has a mixed modality, so it does the taste for the posterior third of the tongue as its sensory part, and then for its motor component, it does the elevation of the larynx and pharynx in swallowing. And then the vagus nerve, I mentioned, it comes out of the jugular foramen and has a mixed modality, and then also in terms of motor, it does swallowing and voice and parasympathetic supply. It has lots of things for sensory. Accessory nerve comes out of the jugular foramen, motor modality, and it causes shoulder shrug. And then hypoglossal, which is the last one, exits in its own canal and has a motor component for its modality, which is tongue movement. So let me just see. So we're just coming up to 10:02. So we can just take a break now till 3 and then come back. And then we'll go through some of the structures in the brain that are the most important, and also the pathways that are most relevant. And in the meantime, a couple of people have written some questions in, so we'll have a look at those and answer those at the end. But if there's anything, just put it in the chat. Okay, hi guys, welcome back. So now we're going to talk a bit about some of the structures. So firstly, the basal ganglia. So these are subcortical structures, which means that they're underneath the main cortex of the brain, which is the outside that grey part. The basal ganglia is made up of the striatum, which consists of the caudate, putamen, and nucleus accumbens. So that's this part here. I don't know if you can see, round. It curves round, and it's got the tail. So the caudate head, body, and tail. The nucleus accumbens right at the front, in front of the caudate head, in red, and then the putamen, which is just underneath the caudate head. Then there's the globus pallidus, which is also part of basal ganglia, and then the substantia nigra, pars compacta, and pars reticulata. The putamen, together with the globus pallidus, is called the lenticular nucleus. So it took me a while to work out what that kind of meant, really. So just that middle part with the globus pallidus, so like a huge, big spherical blob, that is called the lenticular nucleus. And then in terms of connections of the basal ganglia, they're quite complicated, but the cortex projects into the striatum, which is that caudate, putamen, nucleus accumbens, and then the striatum projects into the internal segment of the globus pallidus, and then the internal segment of the globus pallidus projects to the thalamus, and the thalamus goes back to the cortex, and that creates a loop. So I'll just go over that one more time. So the cortex, the grey outer part of the brain, projects into the striatum, which is those three parts, the caudate, putamen, and nucleus accumbens. And then the striatum projects into the internal segment of the globus pallidus, so it kind of goes from out to in. And then the internal segment of the globus pallidus goes into the thalamus, and then the thalamus brings it back round to the main part of the cortex. So that's the kind of loop. And then the conditions that are affected by diseases related to the basal ganglia are Parkinson's disease, obviously, with a substantia nigra, then hemibolism, which affects the subthalamic nucleus, and they get that hemibolismus, which is the movement of the arms, Wilson's disease, which is copper deposits in the basal ganglia, and then Huntington's chorea in the caudate nucleus. So most of these have elements of kind of motor aspects. So the basal ganglia, a lot of it is related to motor and movement. A key structure is the amygdala. So this is a picture with some olives on it, because basically the amygdala is an olive-shaped structure, but also is involved in the hypothalamic control of eating. So hopefully that will help trigger your memory. It's located just above the hippocampus and medially in the anterior temporal lobe, and is part of the limbic system. So the kind of like emotional and fight-flight response is very heavily involved with the amygdala. So there's things like people refer to things like amygdala hijack, or like increased amygdala response. So things like startle, increased fight or flight, they have all relevance to how the amygdala responds to things. The blood-brain barrier is something that comes up a lot, sometimes overlooked. So this is a semi-permeable membrane formed by the tight junctions of endothelial cells in the brain capillaries. And this separates the blood of your normal body from the CSF. And basically there are certain areas which have holes in that allow things to pass through. So larger molecules that wouldn't fit through the normal blood-brain barrier. So they have fenestrations in them. And these are in these circumventricular organs. I don't know what that means, so don't ask me. But these are basically the organs that have the fenestrations. And the most important ones are the posterior pituitary and the area postrema, but also good to remember the subfornical organ, the vascular organ of the lamina terminalis, the median eminence, and the pineal body. So some other important facts about the blood-brain barrier. So as said, large molecules can't cross easily, but also charged molecules, so highly ionized molecules, are not able to cross unless they react with another molecule. Lipid-soluble molecules can cross, but water-soluble ones cannot. And then inflammation causes the blood-brain barrier to become more permeable, and drugs administered via the nose can theoretically avoid the blood-brain barrier because they go directly through the cribriform plate. And then tight junctions are the spaces between the endothelial cells. So these are some other important facts about the blood-brain barrier. Aside from those areas of fenestration, this is also important. So moving on to the white matter pathways. So the central nervous system is made up of white and grey matter. As you know, the white matter is the part that links the brain together. So these are like bundles or wires or cables. And these are kind of divided into these three types of tracts. So the best way of thinking about them is kind of just like cabling between different parts of the brain. So the projection tracts are the cables between the lower parts of the brain, so the spinal cord and the brainstem, to the higher parts of the brain. So they connect the bottom to the top. So if you can just imagine that they're projecting upwards. And then the commissural tracts are the ones that connect the hemispheres together. So in the middle, in between the two hemispheres. And then lastly are the association tracts. So these connect areas within the same hemisphere. So obviously like the right-sided association tracts stay on the same side, on the same hemisphere. So short fibers connect the gyri within the one lobe. So for example, all of the gyri within the frontal lobe are connected by specific association tracts, short fibers within that lobe. And then the long fibers connect separate lobes within the hemisphere. So the cables between like the back to the front, for example, so occipital to frontal, there will be specific long fibers that are association tracts that can connect those lobes, but within the same hemisphere. So just to go over that again, so projection goes from bottom to top, so spinal cord and brainstem to higher parts like the cortex. Commissural connects hemispheres together, so in the middle, and then association tracts are within the same side of the brain, so one hemisphere. Within a lobe, you've got the short fibers connecting the gyri, and then over the whole hemisphere, like through different lobes, you've got the long fibers connecting through the hemisphere. So these are so now that we've understood a bit about what each type is, and I will mention it again because I feel like these are very difficult to remember because you can't really see them, like they're difficult to visualize. But basically the projection tracts, which are, do you remember, projecting from the bottom to the top, so they come from the lower parts of the brain to the cortex and to the higher areas. So the corticospinal and corticobulbar, that's how it sounds. So they connect spine to cortex, and bulbar, like the brainstem region, to the cortex. So that's corticospinal and corticobulbar efferent fibers connecting the motor cortex to brainstem and spinal cord. So projection tracts can also go both ways, so from top down and from bottom up. So those are the projection tracts. And then corona radiata. So these are the fibers to and from all the cortical areas fanning out from the internal capsule. So the internal capsule is actually made from the corona radiata. It becomes that. So the internal capsule becomes the corona radiata. So it's called that because it's like a fan and it fans out. And I've got another picture of it later, but it basically fans out and it spans into all of the cortical areas from the internal capsule. And then the internal capsule is the cerebral cortex's main kind of area where all the fibers connect and link up together. So that's the main conduit of fibers. And the geniculocalcarine tract, which is the optic radiation, connects the lateral geniculate nucleus to the occipital cortex. And that's like a specific projection tract which relates to vision. So the next tracts that we'll look at are the commissural tract. So if you remember the commissural tracts, they connect the hemispheres together. So the projection ones go up and down and connect top to bottom. Commissural connects side to side. And then the last ones, which are the association, connect front to back sort of thing. So with the commissural tracts, the corpus callosum is the largest white matter bundle which connects between the hemispheres. So that is across the middle. And then the anterior commissure crosses the lamina terminalis, and the anterior fibers connect the olfactory bulbs and nuclei, and the posterior fibers connect the middle and inferior temporal gyri. So this is just kind of anatomical landmarks for where those places cross. Again, I wouldn't really stress over learning specifically where those bits are, because you may accidentally take out other relevant information from your brain by trying to learn too many things that are difficult to remember. So maybe just try and remember the names of the main ones and see if you can just explain where they go and where they are. So again, these are quite a lot of tracks which are association tracks. So these are the cablings that connect within the hemisphere. So you've got the cingulum, which connects the front parts of the frontal, parietal, and temporal lobes. So this association tracks connects those parts of the lobes. And then the superior occipitofrontal fasciculus connects the occipital and frontal lobes. The inferior occipitofrontal fasciculus also connects the same, but they're kind of top and bottom. The uncinate fasciculus connects the orbital and inferior frontal gyri of the frontal lobe to the anterior temporal lobe. So this is like a small sort of like horn-shaped thing. And then superior longitudinal, which is known as arcuate fasciculus, is the largest association bundle and connects the frontal lobe to the parietal, temporal, and occipital. That one's quite important. And then inferior longitudinal, which is occipitotemporal fasciculus, connects the temporal and occipital lobes. So as you can see, that within each hemisphere, you've got multiple tracks kind of moving from front to back, connecting both within the lobe and lobes to each other. So gyri within the lobe, but also lobar connections between each other. This is Papez's circuit. So this is a neural pathway involved in processing emotion. And the reason it was important is because it's one of the first descriptions of the limbic system and how emotion can come about. So the circuit connects the cortex to the hypothalamus. So there's a bilateral symmetrical medial surface of the brain, which is where the circuit is. So it starts off with the hippocampus and then moves to the mammillary bodies of the hypothalamus, and then to the anterior nucleus of the thalamus, to the singular cortex, and then to the entorhinal cortex, back to the hippocampus. So this circuit is just sometimes questions are asked about the order of the circuitry. So again, just sort of like a memorization of what order the circuit goes. So we start from the hippocampus, mammillary bodies of the hypothalamus, anterior nucleus of the thalamus, to the cingulate cortex, to the entorhinal cortex, and then back to the hippocampus. And the smiley face is to remind you that it's involved with processing emotion. And then another great, very interesting, easy to remember pathway is the ascending noradrenergic pathway. So we'll just spend a bit of time having a look at these because they can just be a bit complex to remember and think of. So if you think of the locus coeruleus, which is the place where noradrenaline is synthesized and comes from, and you can see it down there in where it says LC and there's a little blob. So that's the blue spot. So it projects in 3 different pathways. And in the syllabus, it specifically says ascending noradrenergic pathway. But there are 3 pathways that actually the noradrenergic system works. So only one of those is the ascending. There's also the cerebellar and the descending. So with descending, that involves motor and sensory functions during sleep and also during wakefulness, and also kind of like all the body systems for noradrenaline. And then the cerebellar system, just as it sounds, just relates to the motor functions and noradrenaline in the cerebellum. And then what they're more interested in is the ascending pathway, which is that long blue line going from the locus coeruleus up into the cortex via lots of different places. So ascending pathway sends noradrenaline to the ventral tegmental area, the substantia nigra, the amygdala, hippocampus, hypothalamus, thalamus, basal forebrain, prefrontal cortex, and sensory cortices. So what's important about that pathway is that the way that it passes through each different area. So when the noradrenaline is in each different place, it has a different function. So for example, in the amygdala and hippocampus, it's involved with episodic and emotional memories. Basal forebrain, it's related to wakefulness. In the midbrain, so the VTA and the substantia nigra, it's involved in wakefulness as well. Whereas in the prefrontal cortex, it's also involved with attention, behavior, cognition. And then when it comes around to the sensory cortices, that's attention and sensory processing. So I would say probably the main point to remember about the ascending noradrenergic pathway is that it's primarily involved in wakefulness, cognition, attention. Also, when it comes up to the hypothalamus, it's involved in the stress response, which is, I suppose, is more what we would think of for noradrenaline. But obviously, it has all of these other aspects as well. And then dysfunction in these pathways may lead to Parkinson's disease symptoms. So I think particularly with the cerebellar aspect, if you're having issues with the noradrenergic pathway in that area, you obviously get kind of Parkinson type symptoms. And we're talking about noradrenaline as well. We're not even on to kind of like dopamine and things yet, but these are the paths for norepinephrine / noradrenaline. And just to remember that dysfunction may lead to Parkinson's disease symptoms. The basal forebrain cholinergic pathway. Again, so with the cholinergic pathways, there's actually 2 pathways, and I'll cover both of them. But in the syllabus, it specifically mentions the basal forebrain cholinergic pathway for some reason. I think the reason is because the basal forebrain is the area that contains the basal nucleus of Maynard. So that's why it probably is more focused on, because that is the site of synthesis for acetylcholine. So the cortex is very sensitive to acetylcholine, and most of it comes from that area, which is the basal nucleus of Maynard. And that area, within that area, the basal nucleus of Maynard is within somewhere called the substantia innominata. So the projections into the cortex are relevant for perception, so in terms of attention, visual perception, and the acetylcholine activates nicotinic receptors in the primary visual cortex. So deficits in the function of the basal nucleus of Maynard are observed in the neurodegenerative diseases as well, such as Parkinson's and Alzheimer's. So because of the investigation into this, they have looked at things in Alzheimer's disease related to acetylcholine, which is why they discovered acetylcholinesterase inhibitors, because they realized that that has an impact on memory and other aspects of attention as well. So what we're really focusing on in this picture is the purple blob and the way that that comes around to the top. So the cholinergic neurons in the basal nucleus of Maynard are projecting round to the cortex, and then we're going to have a look at the green bit now, which is the dorsolateral pontine tegmental constellation, and that's the other area, which is the brainstem cholinergic pathway. So here's just the same image, the same kind of system, but in a different image. So the basal forebrain cholinergic system, that comes from the basal nucleus of Maynard, so which is that's written there as NBM, and that projects round, to the various areas of the cortex in the basal forebrain. And then you've got the brainstem cholinergic system, which goes into the thalamus, to the ventral tegmental area, and kind of around towards the hippocampal area. So other important things to note. So so that lower part of the area projects predominantly into the thalamus. So I think when you're thinking about clinically speaking, I suppose just relevant to remember that acetylcholine is very important for Parkinson's and Alzheimer's, and is reduced in both. And the area, which is the basal nucleus of Maynard, is the most important part for the forebrain cholinergic pathway. So this is also specifically mentioned in the syllabus, the corticofugal glutamate system. But when you look this up, so looking this up online, even in like other notes, like from other places, official notes from like different companies that have got MRCPsych revision sources, there's not really any specific thing that they exactly explain about this. And the best that I could find for this corticofugal glutamate system was that these are the fibers that project from the cortex to the nuclear masses in the brainstem and the spinal cord, kind of as part of the corona radiata and the corticostriate fibers. So I think what it really is looking at is kind of the way that the fibers connect from different areas lower down in the brainstem towards the cortex. And then the other thing to note is that glutamate is obviously the most common excitatory neurotransmitter in the brain. And so as a result, almost all cortical descending tracts rely on glutamate. So there's a large output of corticofugal fibers making up this corona radiata, which are using glutamatergic transmission. So that is probably the link between the two of those things, but separately speaking, there isn't really a particular system known as the corticofugal glutamate system. They're kind of two separate things that they've put together for some reason in the syllabus as a specific thing. So I think just the key thing to remember is that the corona radiata is a white matter tract system, and it uses glutamate to carry information within those fibers from the lower parts of the brain to the cortex. And now we'll talk a bit about the serotonin pathways. So the serotonin pathways, so the largest serotonergic neurons in the brain are the dorsal and median raphe nuclei, which you can see down sort of in the brainstem area, and they're known as B7 and B8. And they provide the main serotonergic input to the forebrain. So they come up from that raphe nuclei to the front of the brain and round into the cortex as well as into sort of the basal ganglia. And then the lower brainstem raphe nuclei, which is the one you could probably see lowest down in those three blobs, that projections into the spinal cord to provide serotonin, obviously, to further down into the body. But the forebrain is mainly supplied by B7 and B8, which are the dorsal and median raphe nuclei. So that's the main thing for serotonin. And now moving on to one of the most important things from basic neurosciences, which are the dopamine pathways, which I'm sure that you've probably revised before or come across before in various different places, but always good to just know it really well for the exam. So we'll just go through each of those pathways and kind of summarize that as well at the end of how to remember those key points. So we'll start with the mesolimbic pathway. So within the mesolimbic pathway, you've got the VTA, so the ventral tegmental area, which is the midbrain, going to the nucleus accumbens at the junction of caudate and putamen. So if you have a look here at the mesolimbic, which is a navy blue, so you can see it starts at the VTA, which is that blob, red blob, and it's going up towards the nucleus accumbens. So mesolimbic is VTA to nucleus accumbens. That is a reward pathway, and the blockade of D2 receptors in this pathway is responsible for therapeutic benefit. So what that means is that the D2 antagonists reduce the positive symptoms of schizophrenia by blockading this pathway. So the mesolimbic pathway is responsible for pleasure and reward. And all you just need to remember is it starts at VTA and goes to NA. So VTA to NA is a mesolimbic pathway, and blockade of this reduces the positive symptoms of schizophrenia. And yeah, so then the next one that we're looking at is the mesocortical pathway. So that's the red one. So as you can see from that, so that projects from the VTA again, from the ventral tegmental area to the frontal cortex. So that is involved with motivation and emotion. And then when that is blocked by antipsychotics, so blockade of D2 in the mesocortical pathway causes the negative symptoms in schizophrenia. And also that leads to reduced activation in the dorsolateral prefrontal cortex and ventrolateral prefrontal cortex and anterior cingulate. And I'll just talk a bit about those. So the dorsolateral prefrontal cortex is responsible for cognition and executive function, and the ventromedial prefrontal cortex is related to emotions and affect. So the thinking of that is that basically because those are responsible for emotions and cognition, blocking that pathway causes the negative symptoms and can cause some of the kind of cognitive symptoms in antipsychotic use as well. So sometimes with antipsychotics, people can have like quite a flat affect, or they can like kind of have reduced cognition or impaired like attention and those kind of things, and that is possibly related to using the mesocortical pathway being reduced. So yeah, so VTA to the prefrontal cortex involved in cognition, working memory, decision making. One note is that amphetamines can upregulate the release of dopamine in the mesocortical pathway, so that increases cognition and activity in the prefrontal cortex. So one reason why people think that maybe amphetamines can sort of make people hyper-focused on things is that there is upregulation of dopamine in that pathway, which increases cognition and activity. And then, yeah, again, so just a reminder that dorsolateral prefrontal cortex related to cognition and executive function within the mesocortical pathway, and ventromedial prefrontal cortex is related to emotions and affect. And blockade of this pathway causes cognitive and negative symptoms in schizophrenia when they use antipsychotics. Next one we'll look at is the nigrostriatal pathway. So that's the light blue pathway there in the middle. So that connects the substantia nigra to the striatum, so the caudate and the putamen. And blockade here causes EPSEs. So what's important about this pathway, so if you can look at the light blue one there, so that's not starting in the VTA, that's starting in the substantia nigra. So these are projections from the substantia nigra pars compacta to the striatum, which is the caudate and putamen. The dopaminergic neurons stimulate purposeful movement, so that is why antagonizing these with D2 antagonists, so antipsychotics, causes extrapyramidal side effects and symptoms, so pseudo-Parkinsonism, because what you're reducing is the kind of purposeful movements, and you're getting these kind of odd and jerky movements because that pathway has had some dopamine blockade. And then the last one that we're looking at of the dopamine pathways, so that is the tuberoinfundibular pathway, which is the light green one. As you can see, it's in a completely different place from the other ones. So that one is the hypothalamus to the pituitary gland, and basically the with the tuberoinfundibular pathway, the hypothalamus, so the arcuate and periventricular nuclei in the hypothalamus, and the infundibular region, the median eminence. So dopamine is released into portal circulation, connecting the median eminence with anterior pituitary, and then dopamine inhibits that prolactin release. So when you block the dopamine, it then causes hyperprolactinemia because you're stopping the stopper. So that is the cause for blockade of D2 causing that. So yeah, so that one goes from the hypothalamus to the pituitary gland, and specifically to the median eminence. So arcuate and periventricular nuclei to the median eminence, and the anterior pituitary. So yeah, so that's quite important just to memorize those for dopamine pathways, because those are kind of the most important ones to know. So that's it for the pathways. Has anyone got any questions at this point? So I know, as usual, I kind of rushed through a lot of the beginning stuff, so I can happy to go through those things. Yeah, so people are wanting to have a look again at some of the hormones. I'll just go back to that. And the white matter tracks as well. I think the white matter track is quite important because it's quite difficult to remember. So the hormones, let's see. Where are they? Okay. Yeah, so again, as said before, like not particularly relevant necessarily to specific brain neurotransmitters, but all of the hormones of appetite are appetite and eating are quite relevant to sort of overall hormonal things, and they all do affect each other. So the cholecystokinin, that stimulates the gallbladder, also the pancreas and the vagal neurons. Then ghrelin was secreted to endocrine cells in the stomach and acts directly on the hypothalamus, and then leptin reduces appetite as well, made in adipose tissue, and then neuropeptide Y itself is made in the hypothalamus, which increases appetite. And I'm not sure if you wanted to also see the other ones, so yeah, so here's the other hormones of appetite. So somatostatin, which is secreted by gastric glands, inhibits release of gastrin HCl, and inhibits release of glucagon in the pancreas, then secretin and gastrin as well. So I hope that is enough of what you were asking. And someone just wanted to go back to the white matter pathways. Yeah, so again, the three types of white matter tracks, so projection, connecting sort of top and bottom, commissural, connecting hemispheres together, association, which is connecting within the same hemisphere, short fibers within the same lobe, the different gyri, and then the long fibers with connecting separate lobes within the hemisphere. So say, for example, frontal to occipital. That is how they would be if you want to look at specifically. So the ones to remember are projection. You've got corticospinal, corticobulbar, corona radiata, internal capsule, and geniculocalcarine tract. So those are the main ones for that. And then commissural, so that's between hemispheres. So that's corpus callosum, anterior commissure are the main ones for that. Yeah, and then lastly, the association tract is probably the longest part of it, which is the different ones that connect within each section. So let's see. Yeah, so cingulum, superior occipitofrontal, inferior occipitofrontal, uncinate, superior longitudinal, which is arcuate, and it's often written as that as well in questions. It's written as arcuate fasciculus, and then inferior longitudinal. So yeah, these are, I know it's probably just like a lot of words and it doesn't really mean anything to you just like that. So yeah, I can understand how it's difficult to learn these ones, but I think just try and maybe separate them in that way. And then someone asked a question about Papez circuit. Yeah, again, this is just the order of the circuit and how it keeps going round. So from the hippocampus back to the hippocampus via hypothalamus, thalamus, singular cortex, and entorhinal cortex, and is involved in processing emotion. Okay, and then another question on okay, hang on. Yeah, so with serotonin, it's getting made from L-tryptophan, which is from the diet, and that can cross the blood-brain barrier. So that goes from L-tryptophan to 5-hydroxytryptophan using tryptophan hydroxylase, and then the L-aromatic amino acid decarboxylase enzyme converts 5-hydroxytryptophan to serotonin. So those are the two main enzymes for serotonin, so tryptophan hydroxylase and L-aromatic amino acid decarboxylase. And then the breakdown, so this is a SERT, like the transporter, and that after that, so monoamine oxidase breaks down serotonin, and then aldehyde dehydrogenase, and that becomes the 5-HIAA. And then those are the things that can block it. Can be blocked by other things, like other medications can also block it, but those are the main ones that are important. So question. Hang on. Yeah. Yeah, just coming back to the someone just wanted to have another look at the basal forebrain cholinergic pathway. Yeah, I think, as said, I think they just picked that basal forebrain pathway because that's where the basal nucleus of Maynard is kept. So I think that's the main reason why it's that one. So yeah, that's it really for that. There's not really anything like particular for, well, I mean, I can, okay, so yeah, that's fine. So the question's about sort of projection into the cortex. So yeah, so it's mainly for attention and for visual perception. So the acetylcholine activates nicotinic acetyl receptors in layer 4 in the primary visual cortex, but also inhibits other population of cells through muscarinic receptor activation, and then deficits in the nucleus basalis, or the basal cell of basal nucleus of Maynard, are observed in the neurodegenerative diseases such as Parkinson's and Alzheimer's, and then that's how one of the reasons why anticholinesterase inhibitors were used for that. Okay, so does that answer the question? Yeah, okay, good. And then the corticofugal, I don't really, it's just a difficult one because essentially the corticofugal fibers are the part that project from the cortex into the masses, like the nuclei, but then the glutamate part of that title, I think, just relates to how it uses mainly glutamatergic it just uses glutamate for its neurotransmission, and maybe that's why it's relevant, because it's the most common excitatory neurotransmitter. So almost all cortical descending tracts rely on glutamate. So that is that. So hopefully that answers the question. Okay, is there any other questions? Or I know like a couple of people specifically had asked for white matter tracks and kind of various other bits for today's session. So if you, I will try and cover like the rest. So I'll just go back to the beginning on the. So when you're looking at the syllabus and you're going through the kind of things that are in the syllabus, so like for example in neuroanatomy, we'll cover the things like the gyri and what happens with each gyrus, like deficits and things, and those types of things in the next one, because that's kind of part of 3 point. Next part, I think it's 3.7 is molecular genetics, and maybe 3.8, 3.9, and 3.10. And then 5 and 6 as well. So there is quite a lot in this section, but yeah, I think it is quite manageable if you're able to kind of split up the time with it. Yeah, so as I was saying, basically, some people specifically ask for white matter tracks and other things. So if there's anything that you really find like you can't get your head around learning it, or you want to cover in the session, just let me know. Send me an email, or you can let me know now, and I can try and include that for the next neuroscience part 2. So we'll be doing the rest of the stuff there in that, and then doing molecular genetics after that. And then those 3 together, those 3 sessions will cover the majority of what you need for basic neurosciences. So yeah, so that's kind of like coming towards the end of the 3rd session. So we've got a session next Sunday, and then there's not one on the 12th of March, and then we return back the following week for the final 4 sessions, and then that will bring you up to the exam. The last session would be the Sunday before the exam, which is on the 12th. So yeah, so we're almost at kind of like I guess just a bit before halfway, really. So you've still got it's about 5 weeks now, 5-6 weeks till the exam. So still a lot of time to get all of this stuff on board, even if you're finding it hard to find time to revise, or there's a lot to cover. But yeah, as said, just if you do find anything that you are struggling with, then just drop me a line. I'll include it in the next session. I've just mainly focused on things that are difficult to remember and always high yield and come up. So there's not any other questions that have come in just now. So I'll stay till 4, and then if there's any other questions, then I'll answer those. But otherwise, I'm gonna go to sleep and get ready for nights again. So yeah, let me know, and let me know how everything is going for you, and any suggestions for things that you might want to look into. Also, the inhaler works great. I feel like maybe I should take it before the sessions, but I think maybe it's like the speaking, it just like triggers it off. But yeah, I don't know. I normally talk quite a lot without problem, but I guess probably after COVID and things. But yeah, so anyway, thanks guys. Thanks for listening, and thanks for your attention. I know it's difficult to keep your mind focused on these topics, but yeah, just a couple of hours every week